Yeah, that's good. So that's the title of the talk, and this is the Molecular Sciences building at Rothamsted. It's been there as a research institute, though not this building, of course, since the 1840s. In fact, the first experiments were started in 1843, and not because we can't think of anything else to do, but we have actually continued those experiments until now. And they all relate to plant nutrition, and the first demonstration that inorganic chemicals could replace farmyard manure. And every 10 years or so, somebody sees nitrate in groundwater and says, oh yeah, we should stop farmers using nitrate as fertiliser, and we should have a nitrogen tax or whatever. And we can use the data that we've accrued over this long span of experiments continuously running to show that, in fact, nitrate can get into groundwater from use of organic manures. In fact, they're worse for it because they go on in the autumn when the plants aren't growing. There's a high rainfall, certainly in Northern Europe, and that can leach out the mineralized nitrogen as nitrate into groundwater. In fact, the groundwater contamination in the UK is still residual from ploughing in grassland in the Second World War to try and make ourselves more self-sufficient. Although it's certainly possible to get nitrate in the groundwater from inappropriate use of fertilizers, we believe that we have to do agriculture with whatever inputs are necessary and whatever inputs can be tolerated, but we should always ameliorate damage to the environment by appropriate use. But now we have a big problem focusing our minds, and in some ways the smallholder farmers that I'm going to be speaking about in Africa, and just to remind you of the billion or so people who go to bed hungry, a very large percentage of that billion are actually smallholder farmers. So these are people who actually farm, who do not produce enough food on a purely calorie basis, let alone nutrition, for themselves. So there's a very big problem there. But they have something in common with the industrial farmer in the prairies here, or in the pastures of Switzerland, or in the wheat fields of the UK. And that is that although we can practice very highly industrialized farming very effectively and produce large yields, we have to use non-sustainable inputs. Firstly, transport in agriculture is a large carbon footprint. But the major carbon footprint, of course, is by industrially fixing nitrogen. The Haber-Bosch process is extremely energy intensive. So we have a town in England called Billingham where we have a very large Haber-Bosch fixing process, and that plant requires more power than the entire town, and it's a big town by our standards. Same is true for Frankfurt and all the sites in the US where you do this. In Africa, there is no real carbon, really no nitrogen fixation process. The plant at Port Harcourt in Nigeria is barely functional, and in South Africa, where the only other plant was, it's actually been sold to China, and it's been transshipped there to go into production there. So there is an issue that we are using non-sustainable technologies which we need to replace, and we have to replace them fairly soon because it's aggravating climate change, which in itself aggravates the problems of agricultural production. And the farmer in Africa, the smallholder farmer, does not buy fertilizer. His economics are such that it's not possible to do that, but also with erratic weather conditions, it's not worth the investment because you may not get a crop at all. And the erratic weather conditions, of course, have been aggravated by the first stages of us accepting climate change. And I've got some slides soon about the long rains and the short rains, which are almost a myth now in parts of sub-Saharan Africa. So this common interest means that we have to look to more sustainability in agriculture in Africa to service people who don't have the access to non-sustainable inputs. But we've also got to do that ourselves. We cannot go forward with the technologies that we've been enjoying to date. And we're going to have to produce more food on a less acreage of available land, and we're going to have to do it more sustainably, if more intensive. And we really do need to help with the poor farmers' situation, but we also need to perhaps learn new technologies which we can access in that process. Okay, right, so that's the background. Now, a lot of the work that I'm talking about is done with the International Centre for Insect Physiology and Ecology. And I knew the people there, based in Africa, with the main lab in Nairobi in Kenya and a field station on Lake Victoria at Mbita Point. I knew them because I'd identified in the late 70s, in fact, a mosquito pheromone. In fact, it's the only pheromone of mosquitoes. And it's produced by all the Culex mosquitoes. And I had very great hopes that it was going to be useful in providing a solution to diseases carried by these particular mosquitoes. They're not the vectors of malaria. They carry filariasis, which causes the symptoms of elephantiasis. And you'll know the genus because it's the main vector genus for West Nile virus, which you have in the US. But what do I mean? Oh, I'm sorry. So a pheromone is a chemical. So I'll repeat your question because they don't hear your question. So the question was, what is the meaning of the term pheromone? So, you know, being English, I use the Oxford Dictionary as my base here. So it's in the Oxford Dictionary. And the definition there is it's a substance produced and secreted by an organism for response, for detection and response by another organism of the same species. It's not quite correct because it needs to emphasize the fact that all kingdoms have pheromones. So I think it actually doesn't say organism, it says animal, in fact. So it's wrong in that sense, in the sense that it's too restricted. So all organisms have the opportunity from evolution of using pheromones by which to communicate with each other. It's even reputed that we have a human pheromone. A trail pheromone. So that's right. So when the worker ant has found a source of food, it lays a trail going back to the nest so that other workers can follow that trail. And that trail is a particular chemical signature, and that is the pheromone, the ant trail finding pheromone. Good example. Sex pheromones are the most kind of popular. And in some shops, you can buy sex pheromones for yourself to enhance your performance at the disco or whatever, but don't rely on it. So this is a pheromone that actually allows the mosquito to say that a site to lay eggs is safe. These are largely urban mosquitoes, and if they lay their eggs in a lavatory which is flushed, then the pheromone is not generated, and so there's no signal lay here. But if it lays its eggs on a safe site, like in this latrine pit, then the pheromone works. So we clean latrine pits and put the pheromone in these little boxes, and the eggs from the mosquito are laid there. And you can get tens of thousands of eggs and located locally. And I had the idea that that could be used then to control these mosquitoes. And I linked up with the Sumitomo Chemical Company to produce a formulation of the pheromone together with a larvicide that would kill. But none of this worked. And it didn't work because you're working in a situation where people don't buy things like we do up here. And health authorities and so on don't have any money to buy stuff like that. And so I realized very quickly that we had to think about different ways of doing things while the rest of the world catches up with our wonderful example of capitalism in the north of Europe and here. So I knew what good people they were. I knew Timothy Onyango was a really good guy to work with. So when I was asked in 1993, when Rothamsted was 150 years old and the Queen came and planted a tree and so on, we decided that we would actively spend more time on poverty alleviation in agricultural communities globally. And myself and one or two colleagues, we chose Africa because we already had connections, as I've just described. And the problem was the stem borer moths. So they come in two sizes. One is the exotic species, like this pyralid moth, Caeloportellus, that followed the introduction of maize into Africa. Obviously, maize evolved in its primary form in the Americas and was transported around the world. It actually came to West Africa via India, and in Kiswahili, one of the languages in the region, it's actually called Mahindi, from India. But the pests followed it, but there were already pests there. Obviously, they weren't pests until they started to eat the crops of this lady here. And indeed, this small grazing is just the beginnings of the problem. As the caterpillars get bigger, they enter the stem or the stalk of the maize and knock the whole thing over. And she will more or less lose her crop as a consequence of that kind of damage. So this guy here from the Gatsby Charitable Foundation, which is charitable money that comes from the profits of the Sainsbury food retailing company in the UK, based in the UK, they were interested in not doing the kind of stuff that our aid agency, which is called DFID, it's like USAID, it's in fact sometimes called UK Aid, but it's really called DFID, Department for International Development. They tend to promote stuff that's already kind of there. So the tools of the Green Revolution, fertilizers, pesticides, and hybrid seed are really what they press out. And it's not worked because it's been out in Africa ever since the Second World War. And although the colonists like ourselves gave way to independence movements, and all the states are independent now, there was a great effort to use these technologies, but they didn't embed. So this guy had the idea that he should come around to leading sites of agricultural research and pick up some new stuff that might be useful in the situation of a growing need for a sustainable intervention rather than just the kind of inputs that we use. And this guy here, he now works for Ceres here, but he was at that time director of the John Innes Institute. It's another institute like Rothamsted, only more at the molecular science. And he was there to sort of advise this guy on whether the science was any good or not. So they didn't have any peer review. It was just the two of them. And they came to Rothamsted, and I said, I think we have a potential solution that doesn't involve external inputs for the stem borer problem. But I want to work with ICIPE, the International Centre for Insect Physiology and Ecology, if I can, because they're people I know. I greatly respect the Director General, Thomas Odiambo. He's a great towering figure of entomology globally and certainly in Africa. There was a very nice person at my talk yesterday, an anthropologist, and she had some stories that perhaps weren't so kind and probably true, but on the other hand, he did a good job in setting up this institute. And he just recruited this guy here, Zia Khan, who was at the International Rice Research Institute in the Philippines. And his job was to try and control stem borers by something that Thomas was calling habitat management. So just as the indigenous pests and the exotic pests had arrived on maize as it had been introduced and looked at as a subsistence crop by smallholder farmers, he felt that if you could manage the wild plants that were around in the agro-ecosystem that the maize was being grown in, then you could possibly prevent the kind of stark damage that that lady on her farm was experiencing. And we had another idea, an idea that came into formal being by a guy at Michigan State. He's still there. His name is Jim Miller. And this idea was to use what he called the stimulo-deterrent diversionary strategy. And there aren't too many people who can say that, so we don't need to engage with you saying it, because you can abbreviate it to push-pull. And the idea I will express now. So we move the idea of habitat management to this formal approach that Jim Miller was trying to develop with horticultural production of valuable horticultural crops like onions and so on. So Zia has continued since 1993 to lead this program, and we are scientific collaborators and help get the international funding. Now, if you don't buy anything, you've got to think of a quite a different entry point to deliver technologies. This is what the farmer's field would look like on a good day. So there's some maize, there's the odd tomato plant growing in between, but mainly it's Phaseolus beans that are growing in there. And they will be harvested before the maize canopy overshadows them, so the farmer will get the benefit of the maize and the higher nutritional value of the legumes. And the farmer, mostly women in the regions we've been working in, will probably try and sell some of the beans, and some of the cobs maybe at harvest. Very, very poor rate of return. But what we've got here is mixed cropping, something you won't see in mainstream farming in the north. And this mixed cropping suggested to us that maybe if we wanted to develop something for controlling the stem borers that would fit in with a no purchasing, no seasonal input agricultural system, we could enter this. And maybe we could pinch some of the land that the beans were on and put in some intercrops that might repel the stem borers. And then maybe around the outside, where they've got these rather weak coffee plants, we could have a trap crop that was more attractive than the maize was. And so that would be a trap crop. And if we pick crops that had some value, that might be a further incentive. So we would deal with the constraints of the moths, but we could also make sure that the farmer wasn't sort of wasting the land completely. We couldn't really imagine doing this with edible crops because they're a very limited range, and they're not normally repellent to herbivorous animals anyway. So we felt that maybe we could get away with using some plants that might work as forage for dairy goats or dairy cows. And that was the kind of plan. So we told this guy Cockcroft that that's what we wanted to do, and that although most of the money would be spent in Africa, it needed to be spent in the ICP lab while we worked out how to do this. And he said, certainly not. You don't need to do any science. You just want to get this push-pull thing going. And we said, well, normally we have a hypothesis, and then we kind of test it and get a plant and that. And he said, yeah, well, this is not normal. We haven't got that kind of money or that kind of time. Dick Flavell says you're really clever guys, so get out there and do it. So we had a council of war with Zia Khan and Thomas Odiambo. We said, but we don't know which plants were going to repel these insects that would be useful in this system. And we certainly don't know which plants would be more attractive. In fact, maize and sorghum seems so amazingly attractive. Anyway, we decided that we would do a completely empirical experiment, which involved triplicated plots at the field station on Lake Victoria of all the forage grasses and indeed many other related species in the Cyperaceae and Typhaceae that would be around in those agroecological systems. And we would pick the ones that were really attractive and we'd try them out as a trap crop. And we'd pick the ones that really had no stem borers ever on them. We'd try those out as the intercrop to see if they would repel the insects. And amazingly enough, very quickly we got such powerful effects that we got on farm. And we started working with farmers almost from the first triplicated plots of plants. And the first push-pull looked like this, but it was a grass that was the intercrop. It was this grass here, Mellinis minutiflora. It's called molasses grass because of its strong smell of molasses, but that chemistry has nothing to do with it repelling the stem borers. And we also had in our mind that if we could exploit stress-related chemistry to repel the moths, because if you're a moth looking for a host, it's not a good idea from an evolutionary or even just a developmental point of view to lay on a plant that's got a lot of your larvae at a higher stage of development than your eggs will be. The larvae will eat the eggs. So we had the idea that if we could get the trap crop, the repellent crop, the intercrop, using that kind of strategy to push away the moths, it might also attract the natural enemies, because the natural enemy is actually looking for a plant that smells like it's got herbivores on it and causing damage. And eventually we moved to this other intercrop, which is one species, Desmodium uncinatum, but the whole genus seems to do this. Not quite as well as the Mellinis minutiflora. We have to use fewer rows of Mellinis minutiflora, but it's best to have 1-to-1 of Desmodium species. But as you'll see, as the years went by, we realized that the desmodium, wherever we used it as an intercrop, was controlling another major constraint. In fact, the other major constraint to cereal farming in savanna farming in smallholder situations in sub-Saharan Africa, and that is the parasitic weed Striga hermontheca. So Striga hermontheca is commonly called the African witchweed. It's a beautiful purple flower, as you'll see, but it's a parasitic weed that can really take out a crop like maize, which is not used to being in an ecosystem where it is, whereas sorghum can manage sort of moderately well. There are other species. Striga asiatica has got bright vermilion flowers, and that's also a problem. That's also controlled by this plant. So it's now 2 intercrops controlling the insects and 1 that controls the Striga hermontheca. And then the trap crop, that was the super attractive plant. We had 2 candidates for that. One was a kind of wild sorghum, Sorghum sudanensis, and the other was a wild but already used as a cattle forage millet, Pennisetum purpureum. So that's what it looks like in diagrammatic terms, and that's what it did for farmers. So we're now on farm with a full push-pull. We actually arrived at a situation initially where we just had the pull. The farmers got the idea of the pull working, and then we foisted the idea of having this push as well. And these are on farm quite a lot later than we started, obviously, and they're on a yearly basis. With 420 farms, and each of the farmer is either very close to or has got a plot where the farmer has been willing to continue with the original best practice. Now, you wouldn't find that in a business-oriented, a capitalist farmer. They will not do that, of course. Why should they? But a farmer who's come into this from a different cultural background, more like we would see a peasant farmer, that has different approaches, and to actually be able to demonstrate the difference between this and that on her farm is culturally something which we never expected to occur, but which is a very popular aspect of this. And throughout, we've got statistically significant increases with the maize in the push-pull fields. And that difference there is a stark difference which allows something like you to go to all your kids at school, whereas no, carry on.
Would you expect to see? It looks like in the first graph there's diminishing, but there's even diminishing of groundwater damage in fields that over the course.
Yeah, it does look like that. Yeah.
Would you expect that to happen?
I don't think we obviously noticed it. It's clever that you did, but we certainly noticed it, and we couldn't really see any particular basis for it. These farms are moderately isolated, but we now have some areas where we've got more or less 90-odd% push-pull farms growing close together. And what our big worry was that we would deplete the biological control agents, the parasitoids. But we don't seem to have done that. We don't think we're depleting the overall population of the stem borers. So I suspect these are just fluctuations. And certainly it's not the same for the striga, which relates to the seed bank. This is much more climatically regulated, but I don't think we can really say anything about it. Maybe one day we might. We'll quote you as an author, okay? And then the yields down here. So you've gone up from around 2 tons per hectare equivalent to more. And that difference there is really what makes this issue of the economy of the farm. But there are other benefits, as you will see, because you have the cattle or dairy goat forage benefit as well. That's now the overall maize yields. These are better now, and these are on a seasonal basis by specific regions. So it's various farms in particular regions. And some of these farms are on the north of Lake Victoria. Some go up into much higher yielding regions of maize traditionally made better, but even where you've got a lower yielding, you've actually got this massive increase here. There's a massive problem of striga in this area, and some very dynamic farmers got involved early on, and they've obviously got it running very nicely. Here, there's less stem borers, mostly striga that we're controlling. So various differences, but nonetheless, a very nice story. A story that was enabling this to be projected at new farmers. So by that time, by the late 90s, the Gatsby Charitable Foundation had agreed that we could actually do some science. So we wanted to find out what it was that was attracting the stem borer moths to the trap crop, but more particularly, what it was that was magically repelling the moths by use of the intercrop part of the technology. So there's a gas chromatograph, there's the chemical detector there, and simultaneously the sample is presented to the antenna of the moth, either as the electroantennogram or as a single cell recording. And I'll probably be talking more about the latter in the talk underpinning the work that I'm going to have on GMOs this afternoon or this evening. So that gave us then the attractants, which were the normal stuff we expect to find in maize that attracts herbivores to it. But the Millennium Award, that's the plant that we were growing as the intercrop that drove away the moths. We were there expecting to find various electrophysiologically active peaks to the attractants, because it's a grass, so it will contain the attractants. But our hypothesis was that it contained extra chemistry which would confer repellency, such that just by being intercropped, it would repel the moths from the maize standing next to it. And so we were able to identify the compounds by the kind of GC mass spec that you do here at Tufts. And one of the compounds immediately explained why this was happening. And that is indeed a typical compound of maize when it's damaged. But it has to wait until the maize is damaged. Now we've got a plant that stuck next to the maize gives the impression that itself, but also the maize, is damaged. And of course, as I said, if you smell like you're being damaged by herbivores and you're a wasp that eats herbivores, that's where you want to be. So we then found, and it was so wonderful we could publish it in Nature with Zia Khan as the first author, that Catesia sesamii and various of the parasitoids that attack the larvae and some that attack the eggs were actually foraging more in the plants where the Millennium Award was. Even 3 to 1 there, we've got a highly significant chain difference in the amount of parasitism there is, even though we've got fewer stem borers as a consequence of the repellency by the Millennium Award. So we've now got the elements of conservation biology working without us having to do anything other than encourage farmers to grow perennial plants in between their crop. And very recently, we found that even the egg laying of the moths, just a cartoon here, causes the plant itself to start to do this. So one day we think maybe we can remove the need for the intercrop, though with it being a useful intercrop, maybe that's not a great idea. But of course it also offers opportunities for the north, because we're now getting a maize plant to do something that it can usefully do in terms of attracting in egg parasitoids and larval parasitoids, anticipating the hatching of the eggs. That's why we call it smart plants. And the interesting thing is, and I'll go to the next slide, because this shows a specific example of a maize plant, and it's a landrace. These are the plants that emerged initially in the Americas from the early ancestors like the teosinte and so on. And with no eggs, that compound top right, called DMNT, is not there, but when it's damaged, or rather when the eggs are laid on it, then you get it. And this is a higher homologue. So these are terpenoid compounds, and you'll remember that terpenoids are produced by carbon 5 units. So if you go up the homologous series from this 11 carbon compound, you get this 16 carbon compound. And these are both very powerful repellents against herbivores and also attractants of the foraging behavior by the wasp. But they're not in the hybrids. So perhaps it's not a bad idea that these farmers are not buying hybrid seeds. They don't do this. Of course, they could buy in some African countries, BT expressing maize. But the BT genes that we have so far from Monsanto, DuPont Pioneer and Syngenta, they don't control the indigenous insects very well. Excuse me. So that systemic effect then is really exciting. So that's now an effect that we're getting distant from where the eggs were laid originally. So that publication there, not as impacting as Nature, but Ecology Letters is quite nice, and it sets you up then to get a grant from the Research Council. That's the UK Research Council, the UK aid agency, now very interested in this work, the Bill and Melinda Gates Foundation from here, of course, from here, and for some other projects in this umbrella of projects, the Government of India. And together with Ed Buckler, who's a maize geneticist from Cornell, and Damaris Odeni, who's from the CGR Institute, ICRASET, we're now working on maize plants that are inheriting from the open pollinated varieties that were created initially from the land races like Cuba and so on. And this farmer, she has actually been looking after these land races. This one is actually called Nyamula, and you can actually buy seed in the market that is produced by a local firm, but it is a land race and it comes from stock from this lady. He's nothing to do with it, actually. He's her husband who doesn't do any farming, but he came in there because we were taking a picture and we just couldn't get rid of him. And we'd promised the lady that she would be photographed for this particular development. And then the work is actually led at Rothamsted, but it's mostly done not just at ECP, but with these people and with the Extension Service in Africa. And the idea is to capture the genetics so that we can help the local breeders make even better Nyamulas and Joeys, another variety, that will do this even more powerfully, even earlier, even more sensitively. And of course, we will patent the genetics so that the patent royalty flow can go back to Africa to improve the work that is done there by these people and the farmers themselves and their extension service. And who are we going to sell it to? Well, we very much like to sell it to the big maize breeders to see if they can rejuvenate their lines and add other dimensions to resistance besides Bt endotoxins, which, as I said, don't control the indigenous pests very well. That's not their problem, but they do have resurgences of other pests like sucking insects. And we believe that this approach will actually control those. In fact, we have evidence for that. And indeed, we have some grants with China to work on this kind of approach against leaf hoppers, brown plant hopper, sorry. Now, the eggs from Chiloportellus, this is the non-indigenous pest, if you roll them back, it's like a piece of plastic. And underneath is the glue that holds it onto the plant. And, of course, it was our hypothesis that that glue would be or would contain the recognition signal that the plant was getting and responding to by mounting its defense and producing that non-atriene compound to frighten away the pests. And so we have been doing fractionated bioassay guided fractionation, and we have several compounds in bioassay back in Kenya. And Samuel Tamiru, who is a PhD student from Ethiopia on the project, when we did the first discovery, he's now a postdoc working on the new project. Has a very nice job, and his family has moved to Beta Point. Right, now the gender balance has changed here because the guys have come out because we've now got some animals and we've got graded cows. And we find that by creating the forage like the Napier grass, which they're learning how to ensilage with technicians from the Kenyan Agricultural Research Institute, who are part of the program, they're very interested because that allows forage to be maintained during the dry season. And while we were talking to the farmers, they really were keen to have a legume back in the system, which we'd kind of pushed out. They're not for every row with the Melanis minutiflora. And this is where we came to put in the desmodium and made this startling discovery that it would actually control striga. And it really controls it incredibly well. In fact, I don't think we would perhaps have realized had it not been such a stark control of the striga. let me just digress a little bit. This is an American charity, Heifer International, and when the farmer can demonstrate that he or she has got enough forage, another guy here with a cow, enough forage for the cow to survive the whole year, then heifer will donate a heifer. And when that heifer eventually calves, the first calf goes back into the system. And they're really well organized. They're all local staff running things, and it works very well. It works so well that the Brits have copied them, and they've got a thing called Send a Cow. So we have memorandums of understanding with these organizations. So as soon as a new push-pull farmer is established, then she gets a cow or a dairy goat. And heifers brought a very good hardy breed of very edible and very high milk yielding goat up from South Africa. And the dairy goats are a real hit. And recently, the farmers are very innovative within the constraints of their system. They've actually got some farms keeping rabbits, which is a really surprising thing to find, but it does really very well on the desmodium mixed with the Napier grass. And some farmers in Uganda with the Send a Cow program are actually farming tilapia with this. I'm not quite sure about the environmental impact of that, but anyway, we will see. Right, so it was the desmodium then that controlled the striga. And this is a very sad and tragic picture that is growing very, very rapidly in Africa. We don't normally fly in. Well, we fly into Nairobi or Kampala or wherever, but we travel around in vehicles. But sometimes when we have distinguished people, and I mentioned the Gatsby Charitable Foundation, so Lord Sainsbury was very interested to see how it was going. So we flew him in to a region of high-density push-pull farmers. And you could see out of the plane window this vast acreage of this parasitic weed. And you know, I said to him that has actually grown enormously since I've been coming to this region, and it's growing far faster than we can keep up with disseminating the ideas of push-pull. So that's what it does. There's a nice looking flower. You see lots of flowers, which means lots of seed heads, lots of seed capsules. So thousands of seeds are created from each flower. And the seeds remain in the soil for up to 20 years, waiting for this poor lady to sow her maize seeds. She's got a small farm of less than an acre, several more kids than the ones there. Husband's died, grandparents still living with her. Very, very typical situation. This is an anecdote, I know, but we have the statistics to show that. And she will really pretty well have lost the crop there. If she gets half a ton per hectare equivalent, she'll be very, very lucky. So the plant has done most of its damage in its subterranean stage. It breaks surface, photosynthesizes, and sets seed. Now, our desmodium was controlling that in a very, very dramatic way. And we quickly demonstrated that it was an allelopathic effect. So another piece of chemical signaling. And this is the main chemical that is doing it. So it's produced in the roots of the desmodium, and it interferes with the way the striga develops after germination. It actually promotes germination to some extent together with the crop. If the chemists among you look at this carefully, they will see a flavonoid, particularly a pigeonin, and then they will see a sugar here, glucose, and they might recognize arabinose, but the link is through carbon. And that is quite unusual, but very typical for these allelopathic compounds, in this case, that do this particular job of killing a parasite that is attacking a different plant. You might say, why on earth is desmodium doing that? You know, why is this altruistic trait in a mere plant? And you can think about that for the discussion. And if you've got a good answer, I'll quote you as an author on that as well, because we don't know, in fact, though I can wave my arms around and say something. And biosynthetically, it's quite interesting. So you might think, oh, well, it's just a pigeonin, and all legumes make a pigeonin. It's one of these nutraceuticals that people search out. And then all you need then is a C-glycosyl glucose transferase and a C-arabinosyl transferase. Well, it doesn't quite work like that. It has to go through this process. It needs that hydroxy group on this dihydroflavonoid ring so that it can open out to accept this unusual carbon-carbon bond structuring and various biochemistry we've done here with colleagues at Rothamsted and ECP. And we now have some of the genes. So we have this gene here. This is the thing that makes the hydroxy group there. We've got it from rice. Rice doesn't make this kind of chemistry, but it does other things that it needs that for. And we've also got the glucosyl transferase that makes this carbon-carbon bond on this thing when it opens out into this keto form as opposed to the enol form here. We don't have that yet, but we will have that from one of the Desmodium species. So that will allow us not necessarily to make a GM maize, but to make an edible bean with the striga controlling trait. And that could be good. And we talked to farmers about it, and they like that idea and would like to use that one day, whether it's GM or not, but it's almost certainly going to be GM. Now, this Desmodium issue, particularly in some of the species which fit more drought-tolerant situations, like Desmodium intortum, works very well in all the other cereals that we've tried. Now, sorghum is indigenous to Africa and has some tolerance to Striga hermontheca, so you can see it growing here, and you will get certainly 1.5 tons or so per hectare here. But where you've got the Desmodium intortum as the intercrop, besides controlling the moths, you've got a much better crop here, and that in this publication is 2 or 3 tons per hectare, and sometimes more in terms of increase. It also works with pearl millet, which is a pennisetum species, and finger millet, which is really hammered, even though it's indigenous, by Striga. Works very, very well there. Rice, Striga, yeah?
Where's Striga native to?
Africa, yeah. So you've got it here, but that's through it being brought in inadvertently. It's easily controlled here by total herbicide. So you let it occupy a trap crop and then take it out with herbicide a couple of seasons, and then usually by crop strength, crop vigor, and irrigation, you would get away from it. It's really a poor person's problem. And that's why it's so tragically growing in acreage there. I mean, it's now in the family Orobanchiaceae. It was said to be in the Scrophulariaceae like Antirrhinums and so on before. And there are a whole raft of parasites there. And in fact, we have a parasitic plant in the UK now called Orobanca crenata, the carnation broom rape. The common name is broom rape. And it's called the carnation broom rape because it has this amazing smell of carnations, you know, the cut flower dianthus. And when this appeared in the farms in Kent, the farmers had no idea what it was because this doesn't photosynthesize. It just comes out the ground like a kind of triffid, you know, and smells amazingly, you know, sort of these crazy articles in the newspapers and so on. And it's moved to the UK from North Africa, where it's progressively wiping out the bean production there. And we were actually benefiting by that because we were selling white beans to North Africa to replace what was lost. But now we've got it ourselves now. And certainly climate change is pushing these parasites around. And there are a number of parasites that you've got in the US that relate to this. So we find that rice, which is not irrigated rice, new rice for Africa is upland rice, and that is also very badly damaged by Striga, but not with the undersowing of a small leaf, with a small leaf Desmodium. The process by which it controls the Striga is suicidal germination. So the germination takes place normally, in fact encouraged by the Desmodium chemicals, but the vascular connection is not made with the plant. And so the seed bank drops off. And we've now got farmers who've got no Striga whatsoever, and though a surprise to me initially, continues. Yeah, Karen.
Just a lot of the comparison graphs have you comparing to maize monoculture.
Yeah.
I'm just curious if you had comparisons with the intercropping that is more common, which it is with.
Yeah, you mean with the beans and so on, like the farmer's practice? Yeah, that would have been a good idea. In fact, there is a small effect of cowpea. So if you grow, I did actually show a slide with some cowpea on it. If you do grow cowpea as the intercrop, you do get a small reduction. So we do have some data like that, but by and large, yeah, this is mostly done on farm, and so it's kind of easier to say, well, just grow the maize there. But in fact, if they do grow the beans, we might not necessarily know, because when we get the data for the yield and the striga count, it's later in the season and the beans have been harvested. So there are some imperfections in this system, but don't forget it's on farm. And that means that somebody's got to ride a bike round to the farm and get the numbers and talk to the farmer and stuff like that. But it's a good point. It's a very good point. We have some problems. The Napier grass started to show symptoms of this strange stunting disease, which is caused by a phytoplasma. We got money then from the Rockefeller Foundation to identify the vector, and having identified the vector, we were able to find races of the trap crop that were resistant. And this is Mr. Ouma. That's one of the technicians on the project. But Mr. Ouma, we've given his name to the one of the resistant cultivars. So it's called Ouma 2. And we have another variety that doesn't have anybody's name on it. But so we're respecting the guy who brought that into being. And we have had problems with drought, growing problems with drought, and with the desmodium not surviving and with damage occurring to the trap crop. So we got a very large program, which is just finished, and we just started an even bigger one with EU funding. And that is to move towards more drought tolerant, not to move into drought habitats, because some of those are semi-natural habitats of importance, particularly for grazing, cattle and so on, but really to help farmers experiencing drought. And we're here using a very interesting trap crop, which is in the Brachiaria genus, which is very extant in Africa, and it's been developed in South America into an apomictic hybrid. So you can actually get non-segregating seed from it. And Brachiaria mulatto 2 is a really good perimeter crop in the terms that I've been describing, but it's also a much better cattle forage than the Napier grass is. And then we've gradually moved to this species of Desmodium intortum and mostly to sorghum in this process. We've looked at a lot more Desmodium species, and we've got a new one that's really incredibly drought tolerant called Desmodium incarnum, and it's got a really high prevalence of this activity of putting in the second sugar, the arabinosyl, and that's what we're using as the material to capture the gene for that. So that's a demonstration plot now of the sorghum, the Desmodium intortum, and this mulatto here has been harvested. It's harvested sequentially. Shouldn't really have harvested both rows, but anyway, harvested sequentially for mixing with Desmodium clippings as the cattle forage. And these are unpublished yields from various trials in some quite degraded land as well as drought-stricken land near to Kisumu, and that may have political impact, as you'll see later. So 100% control of striga and a high level of control of stem borers in the sorghum, and there it is also for maize. I mean, it works really very well for maize, but we're really trying to move to sorghum because of its substantially greater drought. I know there are some varieties of maize that are said to be drought tolerant, but they're not really very drought tolerant. They need quite a lot of water. And that's the take up in Western Kenya. It's very small, extremely small compared with the farmers that could benefit, and the vision even of Kenya from the flying in with Lord Sainsbury. But it's taken a spurt when we've got to the drought tolerant system. And some of these by Alice Muraga are the socioeconomics aspects of that. But with the new adopt from the EU, we have to meet 1 million farmers in the next few years, so that will be quite a challenge. Now, if you go to Africa, there are millions of solutions to their problems, far more solutions than could possibly be managed, and almost 90% of them don't work. And the ones that do work usually involve small bags of seed or fertilizers or herbicide-tolerant cereals and so on, which themselves would eventually have to be bought. Now, that may be okay if the farming is industrialized, but there's a very, very large number of people on the land. And if you industrialize, you don't need so many people. So intensification needs to realistically use this high labor force while things progress in townships and so on to produce more town labor. So this man, Martin Fischler, who's a Swiss guy, and he was a consultant for the Swiss agency BioVision, and they wanted to give a lot of money to this, and he was very skeptical about it. So he came to Africa, and we were giving him the opportunity to visit the farms with us, but he didn't want to do. They wanted to go on his own, and that was facilitated by the fact that to demonstrate that this is real, we've got all the farms geo-referenced. So we know the names of the farmers, and they can be found by the geo-referencing, which you can do very easily in Africa. And so he went around on his own with a little team of people, and to his absolute amazement found that there was a real demonstration of push-pull on the farms. And so he came back, and so we got another raft of money for the extension work in Africa from BioVision and for some other projects. But there are problems. Certainly the seed production. We tried to establish women's groups and encourage farmers to save the seed, and we've tried to encourage small enterprises to buy the seed and sell it on. But we need much more resources and better resources on that. But the seed itself is a commodity. That's the desmodium seed, which the women's groups have actually worked out how to winnow and harvest in a very neat fashion. Extension is very knowledge intensive. And so we have a lot of farmer field days and a whole raft of other approaches. We've got a storyline on the radio. And some of the farmers have put together dramas that demonstrate this show. You imagine the farmer in the States putting on a drama to show how he farms. It's very unlikely. It's even less likely in Britain, because he's more likely to be out shooting pheasants. Not yet, though, but they're pheasants, I said, not peasants. But but these people put on this show, you said. It's a really good show, actually. We've got videos of it, and we've shown them off around. And finally, we have to have a celebrity farmer, of course. As I said, Thomas Odiambo is the founder, and various of the farmers have names which relate to the Lur language, which is the language that Barack Obama's African family would have spoken, are speaking, and the O is related to that fact. And so this is Sarah Obama here, the infamous grandmother of Barack Obama, and in his book he's quite cruel about how she treated him, but I imagine she can see him being some kind of guy who wasn't really going to work hard on a farm. But she's got a reasonably big farm. It's 10 hectares or so, maybe more actually. But she has got some push-pull farming there, and she's very impressed with it. And although she doesn't speak English, she's got a very poetic style that is translated anyway from the Lur. And here she is with Zia Khan and Dickens Nyargol, who is speaking to her in the Lur language about the push-pull operation. This is an anecdote, but it's very important, and everybody believes that having built an international airport at Kisumu now, Barack Obama is going to fly in, and they've got the T-shirt, and we've actually got a lot of farms under push-pull in the very degraded land in that area. So if he ever does make it there, we expect a large input of resources for this program from USAID. But at the moment, it's mostly on malaria. So thanks very much.
You have time for some questions.
I'm going to repeat your question, apparently. The chap over there is going to discipline me if I don't. Is that right? Yeah.
Yeah. Seems, if I understand correctly, push-pull, as you presented it, has two processes. One is behavioral, move into the trap crop, the fusion of the parasites or parasites with lay eggs. But there's also the second process of mortality, egg mortality. So so practices and distinction called push-pull die.
Yes, yes. Well, the question is about the way we describe this simply as push-pull. And since it results in a population control, shouldn't we have push-pull-die? Well, if you go back to Jim Miller, it was the simulo-deterrent diversionary strategy. And the diversion had to include a population controlling aspect of it. So the pull should be where you reduce the population by killing the pests. And that's absolutely right. We just abbreviated it because you know, we have to say this in marketplaces, and the extension people have to say it, and it has to be a sound bite. And of course, there's nothing to do with push-pull in the control of striga, and there's certainly nothing to do with push-pull in providing forage for dairy goats and cows, and the ground being more nutritionally enhanced because of the nitrogen fixation of the legume. So we miss all that, but we've tried it to develop it as a brand name, you know. So what you're saying is absolutely right, and we've been under various pressure to change it, but you know, many of the institutions in the UK have changed their names on a weekly basis. We still like to call ourselves Rothamsted, and it's kind of useful to do that, I think. So we're holding on to push-pull, so I hope you'll help us do that, but it's a very good point you make, and a perceptive one.
So we'll have one last question, and then I want to encourage you, if you're interested in getting involved, to come forward and talk to me individually. But I think I saw another hand. Is there another question?
Yeah, great.
I know you're chemistry, you're a chemist, and obviously that's where you look for your solution in dealing with pests. Is there any information to look for more physical barriers that might exist in the plant that, you know, maybe they're,
Yes. No, I mean, I am a chemist, and you know, I'm extremely excited by chemistry. Still, I still do a lot of compound identification by mass spectrometry, so I was really entertained by meeting Al today. But I have to think objectively, and you know, it's no use me dismissing other modalities. And certainly, in our program generally, we have to consider vision. At the moment, I'm working on an oak decline situation in Europe, where some insects in the Agrylus genus are transmitting a complex of bacteria that causes this decline, we think. And although we're doing the pheromones, we are actually collaborating with Penn State, who are doing the visual cues. So, you know, it's in there. Sound is also another cue. And in fact, in this context, the way that the Napier grass kills the later stage larvae, because it does actually, besides being fed to cows, thereby having them killed, it also kills them itself. That is a physical process, and it inundates them with a kind of gummy latex. Doesn't seem to have any toxic effect other than inundating them. So as they start to form galleries, it's a bit like the need for a mass beetle attack on a tree when you've got the wood-boring beetles, to try and wear out the tree producing resin if it's a pine tree. So that is a physical effect, and you know, I mean, we will exploit the physical effects if we can. Chemical effects are kind of much more amenable to manipulation, we think, and obviously lend themselves to enhancement by breeding, and more particularly GM for the longer term and for the north. So the strategy is that, but I've actually got a couple of papers in press at the moment on changing the color of oilseed rape, that's what you call canola, to purple. And we did it very crudely initially by allowing it to absorb purple dye, but we can now control the anthocyanin pathway in the plant, and then the insects that are adapted to the yellow flowers, particularly the pollen beetles, can't find it. And of course, purple plants are not found by herbivorous birds quite as well. So I do actually have, but it's a good, you should point it out. And I don't think anybody should ever forget that there are an incredible range, as I said, of modalities by which insects and other animals and other organisms interact with plants. And we miss them out at our peril. You may not be able to use them all, but you should know about it.
Well, let's thank John one more time.
take this off?